Hello and welcome to Achievers. In today's video, we are continuing the 5th chapter, Fundamental Unit of Life of class 9th. And today we'll be discussing in more detail the cell organelles. आज हम ज़्यादा डिटेल में स्टडी करेंगे, especially the mitochondria and the endoplasmic reticulum, which are present in cell. Let's begin with the positive thought, which says that your future is created by what you do today, not tomorrow. Fine. So whatever you want to do, put your efforts today so that you can achieve your goals and targets. Fine. Let's begin with the topic. So today we are talking about two cell organelles in more detail. The first one being mitochondria, which is very commonly called as the powerhouse of cell. So let's understand more about it, that what this mitochondria is, how it was discovered, structure and functions. So who discovered this mitochondria? The credit goes to two scientists, which are Fleming and Altman. The Fleming and Altman are credited with the discovery of mitochondria. This mitochondria term was given by C. Benda. Okay, so this term was not given by Fleming and Altman, but they observed it, and later the term was given by Benda. When we talk about the number of mitochondria present in cell, when we talk about the small structure present in cell, and if we magnify it, it looks in a more generalized way of this type. So if we talk about the number of mitochondria which are present in one cell, can be 1000 to 1600, which again can vary from, it can be in some of the cells 1 to many thousands in number, depending on the energy requirement of the cell. If the energy requirement of that particular cell is high, that means the number of mitochondria will also be more in that cell. Fine. Normally when we talk about plant cells have less mitochondria as compared with animal cells, because we do a lot of movement, lot of activities are involved in our day-to-day life, whereas plants have more of a stagnant life, so their cells have less mitochondria as compared with animal cells. When we talk about birds, if I talk about the higher animals and birds, among birds the mitochondria are present in highest number in flight muscle cells. Because you're aware that birds has to flap their wings constantly, and to keep flapping wings, they have to spend energy, and that energy is obtained by a respiration process in the mitochondria. Fine. When we talk about the shape of mitochondria, it can be oval, it can be elliptical, and it can be spiral. So oval भी हो सकते हैं, elliptical भी हो सकते हैं, और spiral भी हो सकते हैं. Fine. You're aware that these are also called as powerhouse of cell. Correct? Or cell furnaces. Like furnaces, in furnaces the fuel burns to give out energy. Similarly, in cells the mitochondria is the place where the food is broken down to release energy. Fine. And this is one of the second largest organelle in the cell. Fine. Let's know a little bit more further about the structure of mitochondria. You can see here I have drawn the structure of mitochondria for you, where you can see that there are two walls. Number one, this one is the first wall, which is called as outer wall. I have labeled it here. This one is the outer wall of mitochondria. This one you can see here, this is the second wall or the inner wall of mitochondria. You can see this is highly folded. This has been made highly folded wall. Now why it is so? To increase the area for the enzymes involved in the respiration process. Similarly, like we have in small intestine the villi, which increase the area of absorption of nutrients. Similarly, here the inner structure is highly folded, so that surface area can be increased. When we talk about mitochondria, we know that mitochondria has its own DNA. So the mitochondrial DNA is present in it. That's why it is also semi-autonomous. It is not dependent on the nuclear DNA to direct it for its functions. It can control its own functions. Why it is so, will be continuing in the video. That I'll tell you that why and how it is helpful. Fine. Now next thing is, when we talk about this high inside this folding of the inner membrane is called as cristae. And when we talk about it has its own ribosomes, which are 70S. This is in the eukaryotic cell, for it is 80S ribosomes are present, but when we talk about the mitochondrial ribosomes, these are 70S. Fine. and the inner fluid which is filled inside, that is called as matrix. Okay, that is called as matrix. This liquid which is filled inside the mitochondria is this. Fine. So this was about the basic structure, and there's one more important thing to know in this, that inside the cell you can see here on this cristae. On these cristae there are some structures like this which we call, which I have drawn for on one or two cristae. These are called F1 particles, and these F1 particles which I have labeled for you here, these F1 particles which are also called as oxysomes, these are involved especially in the ATP production. Okay, this will be having many enzymes which are involved here in the production of ATP. What is ATP? It is एनर्जी करेंसी ऑफ आवर सेल्स. दैट मीन्स हमारी बॉडी के जो सेल्स होते हैं वो एनर्जी किससे ऑब्टेन करते हैं? एटीपी में जो स्टोर होती है एनर्जी उसको. व्हाट इज एटीपी? एडेनोसिन ट्राइफॉस्फेट. जब और एटीपी किससे बनता है? ये बनते हैं एडीपी से. व्हाट इज एडीपी? एडेनोसिन डाइफॉस्फेट. डाइफॉस्फेट मीन्स टू फॉस्फेट्स. एडेनोसिन ग्रुप के साथ जब टू फॉस्फेट ग्रुप अटैच्ड है तो ये है हमारा एडेनोसिन डाइफॉस्फेट. और जब एडीपी के साथ एक और फॉस्फेट ग्रुप अटैच हो जाता है, दैट मीन्स द बॉन्ड फॉर्मेशन टेक्स प्लेस बिटवीन दिस टू, तो बनता है एटीपी. नाउ रिमेंबर कि हमने पहले पढ़ा है, यू हैव स्टडीड कि बॉन्ड फॉर्मेशन में हमेशा एनर्जी इन्वॉल्व होती है. सो व्हेन दिस एटीपी विल बी ब्रोकन डाउन अगेन इनटू एडीपी एंड फॉस्फेट ग्रुप, some energy will be released, and this energy is released in the mitochondria. Fine. And especially in F1 particles. Fine. And there are some processes, some part of this process also takes place in the matrix and some part in the F1 particles. Fine. The more detail we'll study in high classes also. Okay, now when we talk about this structure of mitochondria, we have studied that this is a double membrane structure. ये क्या है? ये एक double membrane structure है. Fine, this is a double unit membrane structure. When we talk about the inner membrane, we have studied that this is highly folded membrane. और ये जो membrane है हमारा, इसको जो highly folded structures बनाते हैं, finger like structures that are called as cristae. जैसे small intestine में villi होती है, वैसे यहाँ पर हमारे पास cristae है. As you see, this can be observed under the electron microscope. Remember कि हम जो छोटे सेल ऑर्गेनलीज है उसको हम कंपाउंड माइक्रोस्कोप में नहीं देख सकते. दे कैन बी ऑब्जर्व्ड अंडर इलेक्ट्रॉन माइक्रोस्कोप. फाइन. एंड व्हेन वी टॉक अबाउट दिस वन cristae, on cristae in especially F1 particles also, there are electron transport systems are present. These electron transport systems are involved with the ATP formation. Fine. And when we talk about mitochondrial matrix, जैसे सेल में साइटोप्लाज्म होता है, वैसे माइटोकॉन्ड्रिया के अंदर जो फ्लूइड प्रेजेंट है उसे कहते हैं मैट्रिक्स. तो मैट्रिक्स, what does this matrix contain? It contains enzymes, it contains ribosomes, it contains DNA, RNA, and as it contains its own DNA, so it is a semi-autonomous cell organelle. Semi-autonomous body का मतलब, जैसे autonomous body होती है, that means it can take its own decisions. Similarly, the mitochondria is also having its own DNA, so it can take its own decisions that what to make, when to make, how much to make, those all decisions. Fine. Next thing is the inner membrane of mitochondria, like we have just seen, contains these oxysomes and F1 particles, and they are associated with ATP formation. And most of this ATP formation occurs in mitochondria. Okay. Now when we talk about the biogenesis, कि mitochondria का जो formation होता है, biogenesis जब बनते हैं mitochondria तो कैसे हो सकता है? Fine. One of the theory which a scientists have given is that they could have occurred, their biogenesis might have occurred from endosymbiotic origin, from sulfur bacteria. Now we are saying that this is one of the inside cell there may be a small sulfur bacteria which has taken the form of mitochondria inside cell. वो कैसा, ये क्या है कि सल्फर बैक्टीरिया की तरह से रिजेंबल करते हैं. और हम क्यों ऐसा कहते हैं? बिकॉज इसमें जो डीएनए में जो माइटोकॉन्ड्रिया में जो डीएनए प्रेजेंट है, इट इज क्वाइट सिमिलर टू द डीएनए व्हिच इज प्रेजेंट इन प्रोकैरियोटिक सेल्स. ओके जो इसका डीएनए है, डीएनए के जो सीक्वेंसेस हैं वो काफ़ी सिमिलर है माइटोकॉन्ड्रियल डीएनए के और प्रोकैरियोटिक सेल्स के डीएनए के. ओके एंड इट इज ऑल्सो सिमिलर इन द टाइप ऑफ राइबोसोम्स. जैसे कि हमने कहा माइटोकॉन्ड्रिया में 70s राइबोसोम्स होते हैं एंड द प्रोकैरियोटिक सेल्स ऑल्सो हैव द 70s राइबोसोम. This 70s is nothing but is a sedimentation coefficient. Okay, जब organelles को study करते हैं when they are put into some machine, then where they are sedimenting उस accordingly unit calculation होता है. So 70s ribosomes भी similar हो रहे हैं. So there are some of the scientists says that this might have occurred from endosymbiotic cells between eukaryotic and prokaryotic, some of the bacterial cells. Okay, moving further. Now let's study in more detail about endoplasmic reticulum, which is called in short as ER, SER-RER, that is smooth endoplasmic reticulum, rough endoplasmic reticulum. Now, who discovered it? It was observed by some of the scientists earlier, but and who was the one who observed and studied in more detail was Keith R. Potter. Potter was the one who had observed this under the electron microscope. Fine. The different parts of endoplasmic reticulum, when we talk about it, can be cisternae, vesicles, and tubules. Now, you might get confused here that its structure is more like this. These same similar structures are present in Golgi body or Golgi complex. Yes, it is similar, quite similar to Golgi complex in its structure, but functions are different. Now, when we talk about cisternae, these are flattened and unbranched stacks, units of stacks. Vesicles, these are oval membrane covered structures, and tubules, which can be regular, these can be often branched structures and which are bounded by membrane. Fine. When we talk about, as we discussed, its structure is more similar to Golgi complex, except now what's the difference here, that in endoplasmic reticulum, these cisternae, vesicles and tubules, they are isolated. They are not together. Like in Golgi complex, you will see the cisternae are attached with the vesicles and tubules, vacuoles and vesicles are attached because they are forming from the Golgi body. But here in endoplasmic reticulum, they are apart, but they are isolated in the cytoplasm. Fine. And they do not form that complex which is normally seen in the Golgi complex. Fine. And second, that Golgi complex is more of a like it is contained in a particular area of the cell. But when we talk about endoplasmic reticulum, it is widespread in the cell. Cell में बहुत सारे areas में होता है, especially the cell which is actively involved in the formation of some of the substances, वहाँ पर endoplasmic reticulum बहुत widespread होता है. Okay, now let's understand कि endoplasmic reticulum का structure कैसा दिखता है. When we look at endoplasmic reticulum, if I have drawn a simple cell for you here, and if you look at the position of the nucleus, as you can see, this is nucleus, and you can see the chromatin material here. So that this endoplasmic reticulum is in continuation with the cell membrane. As you can see here, this is in continuation with the cell membrane, and this endoplasmic reticulum can also be in continuation with the cell membrane or plasma membrane also. Because if some substance has to be made in the endoplasmic reticulum and has to be released outside cell, तो ये endoplasmic reticulum के through होता हुआ plasma membrane से होता हुआ बाहर निकल आएगा. Whatever the vesicles or vacuoles which will be formed. Fine. And that can be present in this way here. Like it can be in continuation here with the cell, and you can draw it here. I may not be able to draw it so well here. Fine. So you can see that endoplasmic reticulum is in continuation with the nuclear membrane on the different sides in the cell, and in this way it form a endoskeleton. एक endoskeleton बना लेता है cell का. Fine. There are several mitochondria which are present in the cell, but you can see them as a very prominent structure in the nucleus. In the cytoplasm. Now, when we look at the further different types of endoplasmic reticulum, this is rough endoplasmic reticulum, where you can see the ribosomes are attached on the outer surface of the endoplasmic reticulum. Fine, you can see these rounded structures. These are ribosomes, and the fluid which is filled inside this area is called as lumen. This area is called as lumen, and these flattened sacs are called cisternae. Fine. When we talk about vesicles, vesicles can be of this shape, tubules can be of this shape, and Smooth endoplasmic reticulum, we call it smooth because it is not having any ribosomes attached on its surface, so it looks smooth from outside. So that's why it is called as smooth endoplasmic reticulum. In short, we call it SER. Fine. Let's understand now what is the difference between smooth endoplasmic reticulum and rough endoplasmic reticulum. अगर rough की बात करें, you are aware that they are having the ribosomes attached on the outer surface. Fine. They mainly have cisternae and vesicles. Where they are involved? They are involved in the protein synthesis. Protein के synthesis में involved होते हैं. And remember, see, whenever somebody asks you that which cell organelle is associated with the protein synthesis, you have to say rough endoplasmic reticulum. It is you should not say only ribosomes, because ribosomes alone cannot make proteins, whereas they are in association with endoplasmic reticulum. When they are attached with endoplasmic reticulum, they are involved with protein formation, but not when they are in cytoplasm and not in association with rough endoplasmic reticulum. Right? Now, where they are found in abundance, that means वो ज़्यादा कहाँ मिलते हैं? Normally in liver cells and in pancreatic cells, they are found in more number. Now, let's understand कि smooth endoplasmic reticulum कहाँ मिलता है ज़्यादा और ये इसका structure कैसा है? You are aware that they do not have the ribosomes attached on them. They are mainly composed of tubules. इसमें ज़्यादा tubules होते हैं, cisternae कम होते हैं. ये abundance में कहाँ मिलते हैं? The cells which are associated with the glycogen and lipid metabolism. जो lipid के metabolism के साथ associated cells है, उनमें ये smooth endoplasmic reticulum ज़्यादा मिलते हैं. Fine. जैसे हमारे adipose tissue which are present under the skin. हमारे skin के नीचे जो fat storing tissues होते हैं, जो adipose tissues इसको हम बोलते हैं, उनमें smooth endoplasmic reticulum ज़्यादा मिलते हैं, या फिर उन लिवर के सेल्स में जो कि ग्लाइकोजन के स्टोरेज के साथ एसोसिएटेड है. फाइन. So these are the two endoplasmic reticulum which are different in their structure and functions a bit. Okay. Now let's understand कि endoplasmic reticulum का overall function क्या है, ये कहाँ कहाँ involved होते हैं. First, as we discussed, it provides the mechanical support and forms endoplasmic endoskeleton. So this endoplasmic reticulum is providing the mechanical support and endoskeleton is formed from it. Second, it is also involved in movement of substances through endoplasmic. That means conduction of substances takes place through endoplasmic. जैसे हमने देखा that this ER is in continuation with the nuclear membrane. So nucleus से जो भी substance का आना है, it can come through the endoplasmic reticulum and come out into the cytoplasm. Fine. The next is, it is a site for rough endoplasmic reticulum is a site for protein synthesis. Protein के formation में involved है. Next is smooth endoplasmic reticulum. This is involved with lipid formation, जैसा कि हमने study किया है, and especially which type of lipids? Phospholipids and cholesterol. Okay. And the next is smooth endoplasmic reticulum is also involved with the detoxification. This is one of the very important functions in our body which is performed by smooth endoplasmic reticulum, which is detoxification. जो toxins हमारे खाने के साथ आते हैं, air के साथ आते हैं, या different medicines जो हम लेते हैं, उनको हमारी body से remove करने का जो important functions होता है, that is done by smooth endoplasmic reticulum. Fine. So drugs, the pollutants, the steroids, they will be removed from our body with the help of smooth endoplasmic reticulum. And smooth endoplasmic reticulum is also involved with the release of glucose from glycogen. Remember, we have discussed here that they are involved in lipid formation. ये lipid formation के साथ भी involved है, और ये glucose का formation from glycogen उसमें भी involved है. Fine. So this is the way we can summarize the important functions of endoplasmic reticulum. These are some of the very important functions. There are several other functions which are performed, but for now, these can be the important ones. Okay, that's it for today. If you have any question, please write to me. If you have not subscribed to channel yet, subscribe it. As you are aware, it serves as a motivation for me, and share it with your friends. Thank you. Take care.